Good afternoon, everyone. I'm glad to be in such a distinguished group of people and working with you in the future. So let me give you the brief overview of GeneCore, and thanks sorry to Ricardo and Philip, because I see quite some opportunities for interactions, let's say developing maybe even new protocols. So there is certainly functional overlap. Okay, what is happening here? Yeah. So clearly, the main application is next generation sequencing, but it's complemented with wet lab support, being qPCR, bioanalyzer, and all what you heard before. But our big part, how we assist to scientific community of not only of EMBL itself, but beyond to EMBL member states, is on this what we call wet lab and bioinformatics consulting, data analysis support. We have worked out genomic data analysis tools which you can view under this geargenomics.com, and as other of our colleagues is teaching and training. Most recently, we are now running the weekly COVID screening for EMBL for all its sites, with exception of EBI. And now we are one of two sites in Baden-Württemberg, which is the southwest state of Germany. And we sequence all positive cases in the genome sequencing, and we can go into that later if it's interesting for you. Now, of course, as others, we are really unlocking the genome with NGS as stated, and ultimately perhaps bridging the genotype and the phenotype gap. And as Ricardo and Philip, RNA-seq in all its guises is probably the leading application. That is also for single-cell RNA. But I would like to mention to you, because I think particularly for this community, it can be really interesting that we are now looking into the possibility to implement this single-cell RNA or transcriptome analysis on bacterial communities. And there are these three concepts out there which have been taken from eukaryotic single-cell analysis to and optimized, or let's say modified for bacterial. And this is a recent development, and I think that it opens a huge space ahead of us and ahead of you. That goes to metagenomics and being either 16 as a typing way, and it can be done with Illumina short-read technology, but also with nanopore. We are now working also in microbiome genomes directly into the cells without any DNA isolation, if those cells are, of course, yielding to its genome. So that has been very particularly economically attractive and also fast. But of course, if you need DNA for whatever further steps, you need not, or you go another way. And then the panoply of other, let's say, under the roof of genomic DNA, studies of chromatin, architecture, methylome, and as I mentioned, also nanopore. So the team is, I think, very, very fruitful combination of a wet lab. You see there are 6 colleagues who are working there. 2 are specifically providing the metagenomic support. I have a person who is kind of on R&D way, and then a dry lab, and who is also my computational assistant, Jürgen, and then this 3 gentlemen who are there to provide support truly on bioinformatics, so analysis of the data. Now, the workflow as such, and I think would like to bring your attention to the flanks on the left and right, where we really spend quite some time in interaction with our users, because we realize the conversation on experimental design and also then the outcome is critical. Of course, there are maybe 2 tracks, how the sample is processed by GeneCore. Either people bring their libraries because they feel, for whatever reason, that may be the better option for them, or they bring the sample to us. Here, again, there's a lot of emphasis on quality control, and we rather repeat a sample than let out inferior results. There is to cope with load, there is a strong emphasis on automation of our protocols. And again, I would say most of the protocols which we are running, or they are demanded by number of samples running, they are also implemented on a liquid handling platform, including the single cell mRNA-seq. And I would like to mention, and that I think is important because you can consider that and get access, our PEPCore, protein expression and purification core facility, has prepared very good TN5, the Nextera, or for fragmentation, and under MTA you can get it too, and it's really making things more economical. In terms of, let's say, particular sequencing hardware, we have 3 NextSeqs 500, which are running to its capacity, MySeq, iSeq, which will retire soon, and we are going to get in next quarter 2 NextSeq 2000. As already Ricardo mentioned, they are providing maybe also higher output if demanded, and it's sitting nicely in the middle between NextSeq 500 and NovaSeq, which, due to diversity of our applications, we are not able to feed sufficiently fast to justify the operation. There's also Chromium, pseudo-pulse field, Telfo, and GridION, because this comes together very neatly for checking of DNA integrity before it goes on nanopore, but it also for PacBio instrument if it gets that. So just to give you the flavor, we are really catering to the community not only of EMBL, but also external users. So this is the proportion of external users last year. And here you see that actually Portugal is sending quite a few samples to sure. EMBL is dominant, as perhaps is natural. Second, our neighbors from Germany, but Portugal is really not far beyond. And I think I would stop here. And if you have any questions, please feel free to ask them. I'm looking forward